Good afternoon, everyone. I'd like to thank the organisers for once again inviting me back to address this conference. It's always a pleasure to do so. I can only apologise, though, this year that I'm not with you in person. I've recently become a grandmother, and at the time of this conference, I am actually going to be in Scotland with my granddaughter. And I thought it best to record the presentation rather than potentially be interrupted by a baby's cries, although I'm sure, being my granddaughter, she won't cry at all. So I've been asked today to talk about forage from the hay biome to horse health. And what I thought I would do actually is tackle this from the horse angle first of all. So I want to look at what we know about the equid microbiome, what problems we know are associated with an unhealthy gut, what management factors actually impact that microbiome, and particularly looking at the feeding and obviously what we're giving the microbiome with different types of forage. So to show that we are all understanding the right terminology that is being used, I just thought I would run through these because I might use them sometimes a little bit interchangeably, but they are referring to slightly different things. So the microbiome is referring to the bugs, so all those live organisms, plus the entirety of the genetic material. So that's inclusive of the plant material and all the excretions. So it's that entire gut environment inside. The microbiota, on the other hand, is the bacteria, the fungi, the archaea, the protozoa, and the yeasts. So it's those live microorganisms when we talk about the microbiota. And the metabolomics, which is newer to this area of research, is measuring the metabolites, the products of this microbial degradation within the gut, and looking at how the microbiota actually influences what's being produced for the horse's utilization. So what do we know so far about the equid microbiome? Well, for a start, we know that it is highly individualistic. We actually seem to see very little diet commonality between horses. So in fact, if you have got a group of horses in a stable and they've been fed the same forage and a similar level and type of concentrate feed, there will still be around about a 52% variation in their microbiome within that population. And Mach found this out when he was looking at 185 horses, so he had a good sample size. What we also know is that the equid microbiome has a rather small and unstable core in comparison with other species. So the human and the monkey have got a bigger core and a more stable core. And the ruminant has a bigger and more stable core again than even the human and the monkeys. So horses are relatively unstable. And this might be one of the reasons why horses are particularly susceptible to dysbiosis and to gut-related health issues. Horses, though, do seem to have a more similar microbiome within the same discipline. So we'll find that a group of dressage horses will be more similar in their microbiome than if you compare them to, say, a group of event horses. But also within that level of discipline, there's a level there where horses that are doing advanced work have a more similar microbiome than if you compared an advanced horse with a novice horse. So that gives us an indication that level of feeding, level of exercise, and probably stress is having quite an impact on the microbiome. It's also interesting to note that compositional differences are seen by the outward and visible signs. So we see that these the fecal pH will be reflected, the microbiome will be reflected in the fecal pH. And indeed, the anaerobic fungal loads will also give you an idea of what's going on in the microbiome. So they can be quite useful biomarkers, if you like. So what about the functions of this gut microbiome? What do the bugs actually do? Well, the bugs determine gut health, digestion, or to be more precise, fermentation. And this is not a surprise. The microbial fermentation of the fibrous part of the feed, the cellulose and the hemicellulose, is vitally important. The volatile fatty acid production from that fermentation can be used to fuel about 70% of the horse's energy needs. But perhaps less commonly known is the influence that the microbiota have on the mucosal barrier. Now, we hear quite a lot about this within human health when we talk about leaky gut syndrome. And horses can also suffer from leaky gut syndrome. And this is when you get breakdown of the mucosal integrity. And in fact, you get the cells separating in little spaces, which allows the passage of material that shouldn't be getting into the bloodstream, because this should be a selective barrier. So things like histamines and toxins shouldn't be getting into the bloodstream. And if they do, they can set up intolerances and allergies. So it's important that the mucosal barrier integrity is maintained, and that is done by certain microbes within the gut. The microbes also kill any pathogens that go in there. So when we look at what's on the feed and the profile of bacteria on the feed, we'll see that some of those, or a high level of those of certain ones, we wouldn't want to get into the bloodstream. So the microbiome acts as a first line of defense there and makes sure that the gut remains healthy and that only the good nutrients and the products of digestion and fermentation are getting into the bloodstream. We know, of course, that the immune health is very related to the gut microbiota. And in fact, 70% of the immune system in a horse is held in the gastro-associated lymphoid tissue. We're also aware now of the gut-brain axis and how this affects behavior. Again, we see this in humans, where humans who have got dysbiosis or problems with their gut, if they do relaxation exercises, they can help to reduce the sensitivity of the vagus nerve, and it may have a significant positive benefit on gut health. We also see this with horses, where if we reduce the overall stress, because you can't actually get a horse to say, well, do some deep breathing, but if you have the environment that is such that the animal can be relaxed and can learn to relax in between times of exercise, then you will have a positive effect on the gut. But what perhaps is less well known is the effect that the microbiome has on the respiratory health in the horse. And we'll come back and look at this when we look at some of the early development of the microbiome in young foals, and it's linked to respiratory health. We also know that the gastrointestinal microbial profile is very region dependent. And this is very important, that those regions are maintained in the way that they should be for good digestive health. So we have an oxygen gradient and we have a pH alteration as we go through the gut. So as you can appreciate, there's a higher amount of oxygen in the stomach and the small intestine, because the food is coming in from the outside. And so the animal's breathing and taking in this oxygenated food, if you like. The pH of the region changes according to where you are in the gut. So you start off in the stomach with a fairly neutral pH, which then goes to a little bit more acidic, and then really quite acidic in the pyloric region, and then moves up to neutral again in the small intestine. And we hope, so that we avoid acidosis, we've got a fairly neutral round about pH 7 in the hindgut, although those two regions are really quite different. So maintaining this grade is very important. So we would expect that the profile is different. However, we do have some what we call keystone species, which are present across the entire gut. So the ruminococcidaceae and the Fibrobacteriaceae are actually present in the stomach, the small intestine, and the large intestine. And they are present in all the different regions of the hindgut. And it's not surprising because horses have evolved to degrade fibre, and we know by some earlier research work that certain more soluble fibres, such as the pectins in sugar beet pulp, are actually degraded in the small intestine of the horse. And for that to happen, there has to be some bacteria there to do that. On the whole, of course, we are hoping to maintain a fairly low level of microbes in the small intestine so that we're actually allowing the horse to do the enzymatic digestion in the small intestine to maximize the breakdown of soluble sugars, proteins, and fats. So the upper tract, though, will contain more facultative anaerobes. So those of the microbiota that are there are things like proteobacteria and lactic acid bacteria, and they, of course, are affected by the feed and by the environment. The hindgut, much higher proportion of fibrolytic bacteria there, and it's in a strictly anaerobic environment, and they are highly pH and oxygen sensitive. And so as the main nutrient source is fibre, this is not surprising that they're mostly fibrolytic activity, but their activity is dependent on a good, healthy anaerobic environment being maintained there. So in terms of what is actually in the foregut, we've got in the stomach, the jejunum and the ileum, we have a variable microbiota, which is highly influenced by the environmental bacteria that is around the horse and, of course, what is present on the forage. We have a high percentage of proteobacteria. These are the gram-negative bacteria. Now, this is a very big and very diverse phyla, and it is influenced by environmental factors. So the type of profile that makes up that proteobacteria is dependent on the environment that the animal is in. The highest abundance in the equine ileum is the pastorelles, and these are also proteobacteria. So it really does depend on what feed is going in there. The firmicutes then, including the lactate producing bacteria, utilize starch, soluble sugars, and any of the readily fermentable fibre, proteins and amino acids, as we would expect them to. And again, very dependent on what the substrate is that's going into the horse's gut. It's interesting to note as well that free-living individuals have a more diverse microbiome than their conspecifics in captivity. So domestication has reduced the diversity of the microbiome. And that is simply because we keep horses on very restricted pastures in terms of the species that they are grazing. So our pastures might include only 2 or 3 species. Some of them even are monocultures of just ryegrass. Whereas out in the wild, a horse being a browser will actually eat a whole variety of grasses from more old and permanent pasture, but they will also browse and they will eat leaves and trees and all sorts of other weeds that we wouldn't normally see in our well-kept pastures. What about then the healthy hindgut profile? Well, generally speaking, a healthy hindgut profile majors on the firmicutes, so it can be anything from 46 to 70%, and that will depend on the type of diet that the horse is eating. And the rest of the microbiome is made up of varying proportions of Bacteroidetes, Verrucomicrobia, Actinobacteria, and spirochetes. So these proportions will alter according to the diet and indeed according to the horse's individual microbiome and what they have grown up with in terms of their early development. But what's interesting to note, and I've just referred to this when we're looking at leaky gut syndrome and that barrier, that epithelial barrier, selective barrier, is that butyrating bacteria are core in all mammals. So they're lactospiraceae. And these have a very important protective function for the colonocytes. So they maintain the integrity of the barrier and stop the toxins and histamines crossing that selective barrier and into the bloodstream. So it just shows how important these different and diverse microbiome is, so that the gut is able to perform the wide variety of functions that it should. So that's what we should have and what it does. So how do we make sure that we can actually have this ideal gut microbiome, if that's possible? Well, the first thing is that you are what you were born with and what you eat. And actually, nature knows best. These animals are born ready to tackle the environment that they come into. So foals are born with a rich and diverse microbiota. The firmicutes are the main phyla, and that's not surprising because foals are majoring on animal proteins and animal feed at this stage because they're living on milk. You do get very large shifts, though, in the first week of life. Not surprising, because, of course, the foal is out of that very protective environment in utero and is being subjected and exposed to a wide range of microbes from the environment and from the mare herself. We get a gradual increase in diversity at 1 to 2 months old, and by 3 months old, the dominant species are still firmicutes, but perhaps not quite so dominant as in the first week of life. But we also have quite high levels of Bacteroidetes, Verrucomicrobia, and spirochetes. The species, that profile remains dominant in older foals. So by 3 months of age, that profile is pretty much fixed. And that's what the animal will recognize as normal in later life. And it is similar to the mare that the foal is with. So a thought here is to be sure that the mare has a healthy microbiome and a healthy gut, and you're feeding her as optimum a diet as possible, because she will influence what type of gut the foal develops. Now, there's been a very interesting longitudinal study which has been carried out by Proudman and co-workers on the early microbiome development and lifelong health of 52 thoroughbred foals that they followed for 3 years, so through some of their early training and into their racing life. And what he found was that high diversity is equal to a lower risk of poor health in later life. And conversely, poor diversity increases susceptibilities to gastrointestinal problems. Well, you would rather expect that, actually, that if you've got a poor gut profile, that you might get more issues of scouring and gastrointestinal problems. But you wouldn't necessarily expect that a low gut microbiome diversity increases the likelihood of soft tissue and orthopedic injuries, not only in foals, but in later life. And that a low microbiome diversity also increases the risk of developing respiratory conditions in later life. And what is also quite interesting about this study is that this impact of diversity happens very early on, and it actually has major impacts quite later on in the animal's life. So if you've got a low diversity at 1 month of age, you get a higher rate of respiratory disease at any time in the future. And this is depicted here in this normal curve, where if we look at the left-hand side of the graph, we see that those animals that are in the lowest 5% of microbiome diversity have actually got a 63% chance of developing respiratory disease by the time they're 2 years old. Now, this is quite notable in the fact that we know that about 80% of our young thoroughbred racehorses are suffering from inflammatory airway disorder. If, on the other hand, your foal is in the top 5% of gut microbiome diversity, then it only has a 15% chance of developing respiratory disease. So those are really quite significant differences in terms of the impact that a good microbiome has on respiratory health. One of the problems that we get with the microbiome diversity is our use of antimicrobials. Now, we do know that foals suffer from E. coli scars, and sometimes you've got no option but to use these antimicrobials. But if you have to, in the first month of life, you get a suppressed gut microbial diversity, and this has a measurable impact in later life. Low diversity not only affects the things that I've just talked about, the gut health, the orthopedic health and the respiratory health, but it also negatively impacts performance in racing thoroughbreds. And this he found through following his 52 foals, looking at the official ratings and earnings. So there's a major incentive there to make sure that the gut is developing well and that you have a good management system that minimizes the chances of you having to use antimicrobials. Because if you do, the likelihood is you're going to have some negative impact on the horse's performance later on. And you're breeding racehorses and keeping racehorses because you want them to perform best. Within the study, and I'm not going to go into all the details, but within the study you can pinpoint certain conditions by picking up certain individual bacteria. So higher Pseudomonas at 8 days old indicated an increased risk of respiratory disorder, and an increased level of Rickenolaceae was associated with gastrointestinal disease. So building a healthy digestive system is of utmost importance, and it's this early colonization, so maximizing diversity, will have a profound effect on performance, metabolic and immune development. And if you disrupt this, you are favoring disease in later life. So you're favoring the fact you're reducing the animal's ability to perform and to be healthy in later life. So that's what happens in our foals. What do we do? Lots of us don't know what's happened to the foals. We buy mature horses. What are the factors that can negatively affect the microbiome in mature horses? Well, just as it does in humans, and there's been quite a lot of press about this, certainly in Europe, about the health of the microbiome and your mental health, the same happens in horses. This is highly influenced by stress. Stress will imbalance the microbiome, and norepinephrine actually has a major effect here, as it promotes the bad bacteria. So it promotes the pathobiome. And so this will cause dysbiosis and issues for the horse, lack of being able to digest things properly. Traveling and stress almost go hand in hand for the horse. And we see that you get decrease in proteobacteria in cecal populations when horses are travelled. And so it's as well to be aware of that constant travelling can compromise the gut microbiome. And you might need to do a few other things, such as turning horses out in the field after a period of travelling, because that has been shown to settle down the microbiome and increase the diversity. For behaviours, so again, we're looking for markers here of compromised welfare and the problems with the microbiome. And for behaviours such as orostereotypies, locomotory stereotypies, aggressiveness and unresponsiveness to the environment, all are indicators of a poor microbiome. There's also a correlation seen between the amylolytic bacteria, so those are the starch degraders, and bowing, which is this rather stressy type of calling that horses do when they're under stress, or particularly when they're isolated. And isolation in horses is equal to stress. But we will also see that they become more reactive, so they'll become just a little bit more difficult if they've got higher levels of a myelolytic bacteria. So this is perpetuated by feeding starch, by poor feeding management. So poor feeding induces microbial changes, and that's not surprising because it's not only what's on the feed, but it's also what microbes it is perpetuating within the gut. So if we add starch to a haylage diet, for example, it's been well documented that that increases the level of what we would call undesirable bacteria. So you get less fibrolytic bacteria and more starch degrading bacteria. And the amount is also critical here, because even less than 1 gram of starch per kilogram live weight per meal can cause a decrease in the core diversity, and this was noted in mares by Venable. Hindgut dysbiosis is seen by an increase in streptococcus, and streptococcus we see in horses with laminitis and acidosis. And when you compare the microbiome of the feces with high forage diets and you introduce starch, you get more pacing in the stable, and you will pick this up. So this is this locomotory stereotypy, and you will pick this up in the fecal microbial profile. But it's not only what you feed, it's also how you feed this. So again, remembering that your horse is a trickle feeder, then it's designed to have a constant supply of food. So 3 small meals per day, you will get a higher diversity in the cecal microbiota than if you feed only 1 large single meal per day. So it's what you do and how you do it is very important. A healthy gut, then, how do we promote a healthy microbiome? Well, haylage only increases microbial stability, and we've known about this for quite a while. We've also known that just hay-based diets and strongly hay-based diets have a higher diversity and richness than if you have high cereal diets. Fibre will also, things that we notice, will reduce stereotypic behaviours, particularly things like crib biting. And that's because if they're continually eating, they produce higher levels of ghrelin, which is the satiety hormone, and then they feel less hungry. But fibre also stimulates gut motility, and therefore they are less anxious, they're less fearful, and they're less stressed. So you've got 2 things going together. You promote a good microbiome, but you also allow the horse to perform its natural behaviours, and the 2 things together will actually promote a healthy gut and therefore a healthy attitude to life by the horses. Manipulation of the microbiome can be used to ameliorate hypervigilance in horses. And we see this where you can do some environmental changes, so make sure that the horse has got constant supply of fibre, and you can turn them out and make sure that they have got conspecifics to interact with. And all of those you will pick up and see a change in the microbiome for the better. So what have we done to date to look at how hay and haylage actually help the microbiota, and how do we get from this healthy food to how does it promote a healthy gut? Well, there are 2 parts to the studies that we've been doing over the last few years. So the first of all is, what is the microbial profile in hay? So what have we got on hay? What are we giving to the horse? And how do our commonly used feeding treatments affect this microbiota? So what does our soaking and steaming, which we do widely to get rid of the respirable dust, but also to reduce the water-soluble carbohydrate levels, what effect does that have on the microbiota? And then the second part of this is to look at the horse factors. So how is the hay and haylage and the microbiota, how is it digested and fermented in the gut? What influence does it have? And also, because of the high risk of changing from one hay to the next, to hay to haylage or hay to silage, we are looking at how this change will actually affect the microbiome and indeed the output, the metabolome. So one of the first studies that we did was mapping the bacterial ecology on the phylosphere of dry and post-soaked grass hay for horses. So we're looking really at what is the profile on the aerial part of the plant, and if we soak it, what actually happens to that profile. So looking at 3 different hays, we had 2 different types of meadow hay, and we had 1 perennial ryegrass hay. And altogether, this is just a graph of the phyla here, but I've only shown the major ones just because it gets a little bit complicated. But we found 28 phyla, 265 different families, 712 genera. The concentration of these was round about 8 colony forming units to the log 10. And between the hays, we had an 81 to 86% similarity. So actually, I think happily for us thinking about choosing hays and what we're looking for, in fact, the differences between hays wasn't that great. But just to look at what we have here now, the large blue one at the bottom is the lion's share of what's there, and those are the proteobacteria, this very large phyla of gram-negative bacteria. Then the next most abundant is the cyanobacteria. Then we have the actinobacteria, the bacteroidetes, and the firmicutes. So we've got a similar range of profile of bacteria there, but they are in different proportions in comparison to what's in the equid gut. Because in the equid gut, we would get a higher level of firmicutes, although, as we've said, in the small intestine, you've got a higher proportion of what is there as proteobacteria. And those 5 phyla make up 96% of what is on the aerial parts, the phylosphere of the plant. So here we have a graph that represents the bacterial families in hay post the soaking treatment. And we'll see on the left-hand side, we have got a representation of one of the meadow hays. So this is on the left, and on the right, we've got the perennial ryegrass hay. And each of the columns represents the treatments. So the left-hand one is the dry hay. Then we've got the 1.5 hours soaking. Next one is 9 hours soaking. And the right-hand column of each group is the 16 hours soaking. And the main families that are present here, without going into the detail of all the lots of individual small proportions of families, we have the Pseudomonaceae, the Sphingomondaceae, Rivulaceae, Enterobacteriaceae, and the Microbacteriaceae. And the P's after each of those represent the Proteobacteria, the C is the Cyanobacteria, and the Microbacteriaceae comes from the phyla Actinobacteria. Now you can see actually how the profiles change a little bit here on soaking. And it seems that we're looking at the meadow hay, that the soaking will increase the proportion of Sphingomondaceae at the 1.5 hours and 9 hours at the expense of the Pseudomonaceae. But when we get to the 16 hours soaking, we get quite an increase in the Pseudomonaceae in that meadow hay. Looking across now to the right-hand side, we have somewhat of a similar pattern, although different proportions in the perennial ryegrass hay. So we've got a smaller level altogether of Pseudomonaceae and a significantly greater level of Rivulaceae and also of Enterobacteriaceae. And then when we move across to soaking for 1.5 hours, we seem to get quite an increase in the Rivulaceae. And then once we get, and same with 9 hours, and once we get up to 16 hours, we get quite a notable increase in the Enterobacteriaceae in that yellow column at 16 hours soaking. Now this Enterobacteriaceae, as I say, it's a member of the phyla of Proteobacteria, and these can contain some notable pathogens. So we've got things like E. coli and Yersinia in there. And so potentially the long soaking does increase the potential pathogens on the hay. Now, if this was a really major serious problem, then also with soaking hay for horses, we'd have a lot more dead horses. But what I'm saying is that horses that have got perhaps slightly compromised guts or a less diverse microbiome might be less able to cope with this change towards these pathogens. And it might be what is causing slight dysbiosis in these horses and lack of performance. So it's worth just noting here that soaking can actually alter the bacterial profile on the forage that you're giving your horse. So in terms of effect of soaking, though, that's just what the microbial profile seemed on these hays. But unfortunately, it is quite variable according to hay type, and the other meadow hay was, as again, a little bit variable as well. So this is very early stage work, and not many people have looked at the microbial profile on forage. But it seems that it is very hay dependent. And this also seems to be the case with water-soluble carbohydrate loss. Now, many other people have looked at how soaking affects the WSC loss. And as you can imagine, that will affect the substrate going into the gut and is bound to have some sort of knock-on effect on the microbial profile within the gut. But looking at the plant itself, why do some hays seem to lose more WSC on soaking and others seem to lose less? And we don't actually have an answer for that yet, because we cannot say that the higher water-soluble carbohydrate content will cause increase in leaching, because that relationship just doesn't exist. But one of the things that we're looking at now, because we're looking at the microbial profile on the plant, is that some bacteria are wetter enhancers, in that they allow the surface of the grass to become wetter, and others will reject the grass, so there would be repellents, and they will prevent excessive wetting of the grass. And it could be the way those bacterial profiles are on the phyllosphere that causes the water to ingress and be able to reduce more of the water-soluble carbohydrates on some hays than on another. And that's a little bit of speculation at the moment, but there is something to do with soaking and why we're getting greater water-soluble carbohydrate losses from some hay and not from others. So this water-soluble carbohydrate difference will undoubtedly, as indeed will crude protein and other effects of soaking, will alter the substrate going into the gut. But will this have an influence on the gut profile itself? And so this is the part 2 of the study, which I'll go on and mention a little bit later. And we haven't got too far down the road on that yet. But to continue our investigation of the pre-feeding treatments, we also wanted to look at the effect of high temperature steaming, because high temperature steaming is becoming increasingly popular, I'm glad to say, because it does seem to have a very positive effect on reducing the airborne respirable dust for coughing horses. But high temperature steaming also has a variable effect on water-soluble carbohydrate and crude protein content, and that could possibly be to do with the bacterial profile on the leaf as well. So we're still looking to see what actually is happening within these different pre-feeding treatments. So one of the things that we know that high temperature steaming does is that it kills a certain level of bacteria. So which bacteria was it killing? And indeed, do we think this would be helpful for the microbiome of the gut? So in this particular study, we wanted to characterize the viable bacterial community of 4 different hays, looking at 2 of the pre-feeding treatments. So what we did was we took dry hay, and then we did high temperature steamed hay. So this is the spike mechanism that really raises the temperature inside the bale to up above 90 degrees and often above 104. And then the third one was to look at just a 12-hour soak, so an overnight soak that somebody might do in the barn. And so looking at the wetting treatment on the alpha diversity, so the diversity within a particular hay, these are the results that we found. Now the top blue line is the dry hay, so that, as it were, is the control hay, and this is what we've got from the environment. And then the grey line is what happens to that diversity as a result of steaming. And you can see here that there's very little variation of the diversity, not of the number, of the diversity of the bacteria species in the hay. However, with soaking, you can see how the diversity is actually decreased as a result of soaking. Now remember, this is not quantity, this is diversity. So what does this mean really? So soaking actually reduced the diversity, as I've said. But looking at the profiles, we actually had an increase in gram-negative bacteria within and between these regimes when we soaked. So what this means is that we've got less diversity there, but potentially we've got more of the pathogenic bacteria as a result of soaking. And this might have an impact on gut health. Soaking did reduce the viable bacteria that are known to cause respiratory disease, and that may indeed be helpful for looking at severe equine asthma. Looking though at the high temperature steaming, that didn't change the diversity, but it did reduce the proportions of microbiota. So in fact, while we still had the same number of different species there, the total number of species were reduced in the steamed hay. And if you've got a fairly contaminated hay, that's a good thing, because we don't want to put a lot of unwanted microbes in there. Steaming also, as similarly to soaking, reduced the bacteria associated with respiratory and dental disease, and both of those things is a positive. But the high temperature steaming also increased the bacteria associated with carbohydrate metabolism, and this we would translate as a good thing, because it reduces the disease risk in equids, because they actually are utilizing more of the feed that's going in there. So looking at the profiles, therefore, that we've got as a result of this, we can see this mirrored in these graphs, where we've got a particular profile on the left for the dry hay, and the middle one is the high temperature steamed, and the right hand one is the soaked. And we can see here that we've got a higher proportion of proteobacteria, and this is the one that contains the potential pathogens. So we've got, we do see that we've got increased levels of firmicutes, which are some of the fiber degraders that we see both in the high temperature steamed and the wet. So looking now, we say, well, okay, this is what we've got on our forage. What difference does it make to the horse? So we're really looking at well, so what? We might be doing all of this, and who cares if it's not making any difference? Do these actually change the function and the metabolite production? So we've gone on now to do a little bit of metabolomics, and particularly looking at volatile fatty acid levels and seeing if we can find differences in the volatile fatty acid profiles. And are these metabolomics influenced by the feed type and indeed feeding practices? Now, we haven't got very far with this, but some of the initial studies, particularly now looking at looking to improve gut health in foals by feeding fibre, we developed an all fibre ration for young foals from birth to weaning, and then from weaning to 1 year old. And this was a very high quality haylage diet, and it was balanced for vitamins and minerals. And we fed this to foals in 2016 to see, A, would they eat it, which they did, but secondly, what would the live weight gains be? And we found they were quite acceptable. Did the foals remain calm? Were they happy on the feed? And what did they look like? And we did find that we had a few pot bellies, and this can be one of the side effects of feeding young foals fibre, because they're carrying too much undigested ballast within the hindgut. So we tweaked the diet a little bit, and to ensure that we get good quality growth, we added a bit of soybean meal, and then we added a probiotic, a yeast probiotic, to help improve the fibre degradation. And we then went on and fed this in a matched pairs trial the following year for 15 weeks. So we had 4 thoroughbred foals fed a cereal diet, and 4 thoroughbred foals fed this new total mixed fibre ration. And looking at the major parameter of the average daily gain, we found that there was equal average daily gain of around about 1 kilogram a day between both groups. So we had equal growth rates on this all fibre ration in comparison to the cereal ration. And it's worthy to note here that the amount of cereals fed was very high. It was close on 6 kilograms per day. But what we also noticed, that there were no joint problems in the total mixed fibre ration group. So those on the fibre ration had no joint problems. Now, at the time, I put this down to the fact that we weren't getting insulin peaks, which you produce on high levels of concentrates, and that can interfere with triiodothyronine and thyroxine, which is very important for the maturation of cartilage to bone. So that's what I put that down to. But now, having seen Proudman's work, it could also be down to just simply developing a healthy microbiome, which in itself might help to regulate the thyroid hormones, but might have another effect over and above that for maintaining joint health. Happily, our developments and additions to the diet meant that we had no pot bellies at all in the second year. And also important was less aggression was seen within the group being fed the all fibre ration. So they were not fighting as much, and while they were certainly exercising and having fun in the field, they weren't kicking each other and being as aggressive, which is always a positive as it reduces injury in the field. So looking then, so that when this was published, we were also looking at the live weight gain, the quality of that live weight gain. So we've got the wither height and the hip height, and we can see that there was no difference between either of the groups. So we're getting good quality growth. But in terms of what we're considering today, looking at the metabolites and the end products of both of these diets, we took feces from these foals, so it was their actual profile, which we used in an in vitro system. And we measured the amount of lactate that was produced, which is on the y-axis here, and the x-axis is the incubation time. So we ran an incubation from the start through to 62 hours, and we started to take samples for delactate production. And the yellowy orange line at the top here shows the lactate levels that were in the bottles that were inoculated with cereals. And the green line is the one that is from the bottles that had the fibre ration. And you can see how much more lactate was actually produced as a result of feeding cereals, and how in fact the lactate levels on the TMFR diet, the fibre diet, remained fairly low and constant throughout the incubation period. So this actually shows that how you can influence the microbiome and actually the products of this fermentation as a result of what you are feeding. Now, we don't want too much delactate because that causes acidosis and other issues, and as I say, it's indicative of high levels of insulin as well. So this work did show us a little bit about what is going on in terms of the output as a result of changes in the microbiome. So the rest of our stage 2 work is ongoing at the moment, and in fact we're going through looking at a lot of data. But we've done an experiment with 4 different haylage diets, and we're looking at the differences in apparent digestibility, the fibre fractions, and the pH of the forage versus the pH in the feces, and how this relates to the gut metabolomics. So how does the apparent digestibility give us in terms of the profile of these products of digestion. So on each diet, but also at that critical changeover from one haylage to the next, because we know that a high level of colics occur when you swap a horse over from one fibre diet to the next. So we wanted to know how quickly any changes in the microbiome take place and if they actually are reflected in the metabolome, and to see if we've got any apparent digestibility and metabolomic association. So just to give you a hint of some of the results here, we have got the younger haylage, as you would expect, so that of lower physiological age and higher leaf to stem ratio had a higher apparent digestibility. We did find slightly lower fecal pHs on the wetter haylages, so those of lower dry matter did drop the pH of the feces a bit. Overall, the profiles of acetate, propionate and butyrate in the feces was influenced by the different haylages. Looking then at the metabolomic differences once the animals became stable on the diet, we found that we couldn't detect any differences in urinary metabolites, and either they're not seen there or the particular ones that are in the urine weren't actually influenced. But we did see some differences in the fecal metabolites. So in some of the major ones like the acetate, the malinate, the methylamine and some of the other minor metabolites, we did see differences between different haylages. Now at the moment we don't know if this is down to the substrate nutrient concentration, so what's actually getting into the gut, or if this is a little bit more dry matter driven, but the two things go hand in hand. So something is actually influencing the metabolites. So we are getting a change in the end products of digestion. So in conclusion then, the microbiome influences the whole body, and that's a very important factor to remember. The gut, the immune system, the respiratory system, the orthopedic health are all determined by how healthy that microbiome is, but also the athletic performance. And if anything is an incentive to look after the microbiome, it is the fact that you'll get a better performance from your horse if it has got a healthy gut. So consider how the management affects this microbiome. The feed, nutrient and hygienic content is very important to the development and the maintenance of that microbiome. But as indeed the feeding schedule, how often do you feed? Are you feeding to maximize that throughput and maintain that very useful gradation of oxygen and pH down through the gut? And therefore you could consider very carefully what you should be feeding and find ways of utilizing those substrates and high quality fiber diets that we know help to produce a healthy microbiome. So my main message today is, if you look after the microbes, then they will look after your horse. So I'd just like to thank my co-workers for their many hours in the lab and work and help with these experiments, Dr. Simon Daniels and Sue Coe-Martin. We had some very good PhD students that helped us with some of this work, and also our two sponsors, Eclipse and Buckland Feeds. So thank you very much for your attention today, and I hope that you have found that interesting, and I hope you enjoy the rest of your conference.